Hello and welcome to this video tutorial on oxidative phosphorylation, which is the final stage in aerobic respiration. Now the phosphorylation refers to the addition of an inorganic phosphate ion to ADP to give ATP, and this is connected with the oxidation of molecules. Now I'll explain that in more detail later on when you see it, see the actual reactions involved. If you want to know more about ADP and ATP, and particularly the structure of ATP and its role as a short-term energy storage molecule, then I do have another video on YouTube called Introduction to Respiration, Glucose and ATP. So what happens in the overall process of aerobic respiration is that the chemical energy stored in molecules such as glucose, this is also true of fats by the way, but at A-level biology you really just look at glucose. The chemical energy stored in this molecule is released during its dismantling, and at the end of that dismantling process, the very end of respiration, all of the carbon atoms, of which there are 6, they finish up as carbon dioxide. So the carbon atoms have combined with oxygen, and similarly the hydrogen atoms, they finish up being water. So these are the products of respiration. If I write the overall chemical change as a balanced chemical equation, you have glucose C6H12O6 as we see here, and it's going to give carbon dioxide, and because we have 6 carbons, that's enough to make 6 molecules of carbon dioxide. 12 hydrogen atoms is enough to make 6 molecules of H2O, and you see straight away that on this side we have altogether 18 oxygen atoms, but only 6, here they are, in the glucose molecule, so we need some additional oxygen, and this is the oxygen that we breathe in through the lungs. So that's the overall chemical change of respiration, and of course that is associated with the release of energy. So it's an exothermic reaction, and there's a lot more about the energetics of this in the video on ATP and glucose. As I mentioned in that video, the overall chemical change that happens in aerobic respiration, it's no different to that for the combustion or the burning of glucose, in which case the energy is released as heat. In respiration, we don't really want to release the energy so much as heat, but we want to capture it and store it as ATP. So out of one glucose molecule we can get anywhere between about 32 and 36 molecules of ATP. Now here it looks like the energy is released in one go. In fact, most of the energy is released really at the very end, and it occurs when the hydrogen atoms that we've removed from glucose combine with oxygen to make water. Carbon dioxide production is just a way of getting rid of these carbon atoms, but most of the energy is released when these hydrogens become water, and that occurs at the end of the so-called electron-transport chain. Now we'll just take a closer look at the combination of hydrogen with oxygen to make water. If I just put it in words here, hydrogen plus oxygen plus, goes to give water, and you will know from GCSE science or even Key Stage 3 that this releases lots of energy. This is of course the squeaky pop test for hydrogen gas. If you have a test tube with hydrogen in, you put a glowing splint in there, it reacts with the oxygen, and you get this squeaky pop as the energy is released in the form of heat. Now if I write a chemical symbol equation for this, hydrogen is H2, water is H2O, and oxygen is O2. Now we see in oxygen it's diatomic, so we get 2 oxygen atoms in each O2, so that's enough to make 2 molecules of H2O, but then we need 4 atoms of hydrogen, so each H2 that we have here consists of 2 atoms, so we need 2 molecules of hydrogen. So we'd normally, instead of writing it like this, we would just use the balancing numbers, so it would be 2H2 plus O2 gives 2H2O. So that's the reaction of hydrogen gas with oxygen to make water, and we have the release of energy. Now in the mitochondria, electron-transport system, at the very end of respiration, although the oxygen involved is present as O2, the hydrogen certainly is not. This hydrogen is the hydrogen that's removed from glucose or other fuel molecules. It never occurs as H2 gas. So for the moment we can write it as hydrogen. That's to represent a hydrogen atom, but we're not really saying exactly the chemical form of it. We need 4 of them plus oxygen to give 2 H2O molecules. Now, in order to capture the energy that's released from this reaction in respiration, this is broken down into lots of small individual reactions. These are the reactions that, as you will see, they take place on the electron-transport chain on the inner membrane of the mitochondria. I'll show you all that later. But the point is, there isn't a simple one-step reaction as written here. This is broken down into lots of little stages, and that involves, first of all, dismantling the hydrogen atoms into their electrons and protons. So if you remember from GCSE chemistry, I'll put this up here. If we think of a hydrogen atom, we have a proton in the nucleus and 1 electron in the first shell. So the hydrogen atom is the simplest possible atom. There it is. And we can think of that as being a proton and an electron. In fact, during respiration, all of these hydrogen atoms, they become separated. The electron gets taken from the atom, so all that we've got left is a proton. There are no neutrons in a hydrogen atom. So sometimes most of the time you'll see that just written as H plus an electron. Okay, so that's the way to think of hydrogen atoms. Think in terms of the individual electrons and H plus ions. Now you will see that referred to as a proton or as a hydrogen ion, or just as a H plus ion. Now, although the hydrogen ions occur in lots of places in respiration, they occur just dissolved in the water where they're associated with water molecules. If you do any A-level chemistry, you know that free H plus ions don't really occur, they occur bound to water, but if you're doing just biology, don't worry about that. The point is there are plenty of H plus ions around in cells, whereas electrons never occur as free electrons. So these electrons that get taken off the hydrogen, they're always being given to something, so they'll be given to an electron carrier. So that might be a metal center in an enzyme, for example. Again, don't worry too much about that, but it will all be clear to you later. So if we go back to our reaction where we're making water, H2O, in the final stage of respiration. That's I'll write that above now, because in the final stages you've got 4 hydrogen atoms plus oxygen. We're going to give us 2 molecules of H2O and some energy, which is used to make ATP. Instead of writing 4H, we can write these. Each hydrogen can be an individual proton and electron. So here we can just write H plus plus an electron. If we've got 4 atoms, then we've got 4 protons and 4 electrons. They combine with oxygen to give 2 H2Os. And that's what really happens on the inner membrane of the mitochondria. Although overall we're transferring 4 hydrogen atoms to an O2 to make 2 waters, they travel, if you like, independently. With this diagram I would like to just give you an overview of how a molecule of glucose is dismantled as it proceeds through glycolysis, the link reaction and the Krebs cycle. The main thing to appreciate is that the carbon atoms in glucose, of which there are 6, they are released in the form of carbon dioxide gas, and the hydrogen atoms, they are released at various stages, and I'll say how they are released and where they go in a moment. At the end of glycolysis you get 2 molecules of pyruvic acid, and each one has still got 3 carbons, so we've not actually lost any carbons at that stage. Now you will hear that called pyruvate. That's just the ionized form of the acid. If you're not doing chemistry, it doesn't matter, you can call it pyruvic acid or pyruvate. But the point is you get 2 pyruvates. Pyruvic acid has only 3 carbons in. So you have the link reaction happening twice, which is why I've got 2 carbons. In the link reaction you get 1 CO2 released and 1 hydrogen is transferred off the pyruvic acid. So that's why I've got 2 here. Similarly, the Krebs cycle goes around twice because you have 2 pyruvic acids going into it, and that's why I've got 2 of all of these. So if you look overall at what happens, you've got 6 carbons, 2, 4, 6, and you've got 12 hydrogens, 2, 4, 6, 8, 10, and 2 of them in glycolysis. Now superficially this all looks nice, everything's balanced up, but in reality we would need to look a little bit deeper at these hydrogens. We would have to take into account the fact that a hydrogen is really a proton and an electron, and many of the protons from these hydrogens, they get swapped with protons in the surroundings, so on other molecules, on ions and on enzymes. So to balance the protons, the full hydrogen atoms, it's a little bit more complicated than this, but if we think of each of these hydrogens as being 2 electrons, then it does work. Okay, so you've really got to think about the electrons in here. That's beyond what you need to know at A level, and I may well make an extension video on this, particularly for undergraduates, but all you need to be concerned about at A level is that the whole purpose of these reactions is to release the carbons from glucose as CO2 and the hydrogens, which are going to find their way to oxygen on the inner mitochondrial membrane using the electron-transport system. The glycolysis is happening in the cytoplasm, the Krebs cycle is happening in the matrix, and these hydrogens are going to go then to the inner membrane and release their electrons into the electron transport chain. We need to look at how they do this. How do these hydrogens come off? How do they come off the molecules in the Krebs cycle and get to the inner mitochondrial membrane? Well, we have these couriers of electrons, or carriers. One of them is called nicotinamide adenine dinucleotide. So it's often abbreviated, well, this is to NAD, and the other one is flavin adenine dinucleotide, abbreviated to FAD. Now, all you need to know for A level is that FAD and NAD, they are small molecules, and we call them coenzymes or even cofactors. The small molecules that are necessary for enzymes, certain enzymes, to be able to function, and their role is that they transfer, or they carry hydrogens. Now strictly speaking they carry electrons. In fact it's a bit more complicated than that. We'll see that in a moment. If you're doing A-level chemistry, this is a hydride carrier. It's equivalent to the, it carries the hydride ion that you use when you're reducing aldehydes and ketones, for example. It's the biochemical equivalent of NaBH4, the sodium tetrahydroborate 3. But that's enough chemistry for now. Easier way to think of these is these are couriers. They are the white vans, the FedEx Express or the DHL, that deliver hydrogens, okay, from the glycolysis and link reaction and Krebs cycle to the inner mitochondrial membrane. Now, to look at how they do this, because a lot of people get very confused about how do you abbreviate, what's the proper symbol for FAD and for NAD. Well, I'm going to start off with FAD because that one is much easier to understand. FAD can be given 2 hydrogens, and we write it as FADH2. Now, you would call this the reduced form of FAD, and you would call this the oxidized form of FAD. And in your exams at A level, it's fine just to call that FAD reduced. As long as you're clear about what you're saying, you could write FADH2, or you could write just FAD but say reduced form, something like this. This is a reduction reaction. We're adding, when we add the hydrogens, there's some people not doing chemistry, they get confused about reduction and oxidation. Reduction is always when something gains electrons. And to see why that's the case, we have to remember that when we've got 2 hydrogen atoms, what we've really got are 2 H pluses and 2 electrons. And it's the fact that we're adding electrons to the FAD that it defines this as a reduction reaction. The protons, the H plus, they just happen to go along with them. They're just going along for the ride. So I know at GCSE, oil rig is what you use when you're doing your GCSE chemistry. Oxidation is loss, reduction is gain. This always refers to electrons, and so sometimes it's free electrons, but sometimes it's electrons that take along their protons, and so it's a whole hydrogen that's transferred. And of course this reaction is reversible, so you can take the electrons off, or the hydrogens, and go in reverse. So FAD can collect the hydrogens from, well, it collects them from Krebs cycle actually, and then it forms a reduced form, and then it can release them on to the electron carriers of the inner mitochondrial membrane. So strictly speaking then, if we want to write a correct equation for this, we would write FAD plus 2 electrons plus 2 H plus, and it's a reversible reaction, and it gives us the reduced form FADH2. So that would be a sort of a correct version, a bit more information than just writing a hydrogen in square brackets. Now it's a little bit more complicated with NAD, and a lot of my students, they say, oh, the teachers have told them how they should and shouldn't abbreviate NAD, and I hear all kinds of stories. The thing to remember is that NAD, in its oxidized form, it's actually got a positive charge. Okay, so if we add to that the first electron, just add 1 electron to that, it cancels out the positive charge and it gives us NAD, okay, plus a negative neutral. And then if we add a second electron, it would give it a negative charge. So NAD with a minus charge. And then if we add a H plus ion, a proton, we get NAD with a hydrogen, but there's no charge because the positive and the negative cancel out. Now that is what we call reduced, the reduced form. So again, you can write it as reduced NAD if you're very clear, or you can write it NADH. They both mean the same thing. This is the oxidized form, and to be honest, at A level they're not looking for that positive sign at all. This is just to help you make sense because you may be confused about this. It's perfectly fine to write NAD and without a charge and NADH, or to write NAD oxidized and NAD reduced. I don't like writing NAD oxidized because that means you're actually taking electrons out of NAD and making it into something else, but you know, let's not go there. But if we now look at what's happening, what have we given to this NAD when we've reduced it? It's a reduction reaction because we're giving it electrons, okay? So we've given it 2 electrons plus 1 H plus. So if we write this down as an equation, we've got NAD plus, we're going to give it 2 electrons, there they are, and we're going to have 2 protons there because if we've got 2 hydrogen atoms, then in 2 hydrogen atoms you've got 2 electrons and 2 protons. So what happens is, and I'll put this as a reversible, is only one of these protons goes on, so you get NADH, and the second proton just hangs around, it just stays in solution. Okay, so strictly speaking, NAD is a carrier of a pair of electrons and 1 proton. In chemistry that would be a hydride ion. Effectively it's a hydrogen atom that we've put an extra electron on. Okay. Now is a good moment to consider the structure of mitochondria. So this is a diagram of a single mitochondrion. It has 2 membranes, an outer membrane which is relatively permeable, so molecules and ions can pass between the cytoplasm and the space between the 2 membranes, which is the intermembranal space, whereas the inner membrane is not permeable or not as permeable as the outer membrane. It's selectively permeable. It exerts a lot of control over the movement of ions and molecules in and out of the centre of the mitochondrion, which is called the matrix. Now the inner membrane is folded into these sort of finger type projections which are called cristae, and that increases the surface area, and it's on these cristae you have these little stretches where ATP is made, and that is the site of oxidative phosphorylation. So you've got the electron-transport chain is located on the membrane, as well as the basically the ATP synthase, the enzyme complex that makes ATP. This is the image of a section of heart tissue seen through an electron microscope, so it's been magnified 30,000 times. These lines here, these are the protein filaments responsible for contraction of the muscle, and you always get lots of mitochondria in muscle tissue because the contraction process requires a plentiful supply of ATP. Now you can see very nicely on these mitochondria that they have a double membrane, the outer and the inner mitochondrial membrane, and you can see also the cristae, these finger-like projections which come into the matrix. So far then we can summarise what has happened. The glucose has been dismantled through the reactions of glycolysis, the link reaction and Krebs cycle. We can think of the carbon atoms in glucose now being in the form of carbon dioxide. It's strictly speaking not the same carbon for carbon, but for every carbon that is in glucose you get one CO2, but there is some exchange with other molecules, but you know, the equation balances, you don't want to worry about the details there too much. The hydrogens, they have been plucked off the glucose and collected by NAD and FAD. From each molecule of glucose you get just 2 molecules of reduced FAD. This occurs in the Krebs cycle, but you actually get 10 molecules of reduced NAD. So this is the major route for the hydrogens. It's on the NAD rather than the FAD. The point is, these couriers of hydrogens, they collect the hydrogens and they deliver them to the electron-transport chain on the inner mitochondrial membrane. This is where we have to think of the hydrogens really as individual protons and electrons. So a hydrogen atom, we have to, as we said earlier, have to remember that it's a proton and electron. So when these reactions take place in the electron transport chain, the protons and the electrons are separated, make their way to oxygen independently, shall we say. Oxygen is going to use the hydrogens to become H2O. So there's a H2O. And as we saw earlier, because we've got 2 oxygen atoms here, that gives us 2 molecules of H2O. So we need, in fact, 4 hydrogen atoms. So there they are. Now each FAD in the reduced form or NAD in the reduced form delivers just 2. So we have to have 2 of these carriers giving up their electrons or being oxidized for every molecule of O2 that becomes H2O. So in the case of NAD, I can write 2 NAD in the reduced form with the accompanying 2 H plus ions. They react with oxygen and that gives us H2O, 2 molecules of H2O. So that's the oxygen. I think I need 3 hands here. 2 of those plus 2 NADs in the oxidized form. So I'll just check that. 2 pluses on that side, 2 pluses on that side. So you could just write NADH2 if you wanted, and NAD there at A level, that would be fine. That's the overall change, but it doesn't happen in one step. It's broken down into lots of little steps, and this is how the electron transport chain manages to capture the energy released. That is a reaction that gives out enough energy, in fact, to make 7 ATPs, but in practice we only get about 2.5, and that's achieved by breaking it down into lots of little steps, which is what we're going to look at in a moment. And just to show you the equivalent reaction with FAD, so it's a little bit simpler. The point is you need 2 of the reduced FADs, so they're bringing altogether 4 hydrogens, and there's the oxygen that's going to be reduced. So if I put that one over there to start off with, so we get 2 molecules of H2O and we get 2 molecules of FAD. In order to understand how the hydrogens from reduced NAD become the hydrogens in water by means of the electron transport chain on the inner mitochondrial membrane, this energy level diagram can be helpful. You can think of the electrons in NADH being of higher energy than those in water, in the same way that water in a reservoir up a hillside has more energy than water down in a lake at the bottom. So when we send electrons from NADH onto oxygen to make water, it's as though we're letting the water come down a waterfall. So if I open the gate here and allow the water to flow down the hillside into this tank at the bottom, or lake or reservoir, it's going from a position of high gravitational potential energy because of its height to low gravitational potential energy, and the energy is being released, well, it's been converted to kinetic energy as it's passing down the hillside, and then the water is transferring that energy to the surroundings. It's colliding with molecules in the air and giving them kinetic energy. So it's really a heating effect, and you have sound energy as well. That's the vibration of the particles in the air. And so by the time the water's got to the bottom, it's lost some of the energy that it had when it was higher up. And it's a similar thing that happens in the mitochondrial electron-transport chain. As the electrons go from reduced NADH onto oxygen to become electrons in water, energy is released. Now we could just release that as heat, but we're going to convert it into ATP in the same way that if we wished we could put a little turbine on here, so it's really just a water wheel, and so we could make the water wheel rotate, connect that to a generator and produce electricity. So that way we've harnessed the energy that's being released as the water comes down the waterfall and transferred it into electrical energy. And this is what we're doing here, but it's on the electron transport chain. We're harnessing the energy and we're going to store it temporarily in another form, and that form is a gradient of hydrogen ions across the inner membrane. I'll explain that in a moment. Here we can see for the first time the actual electron carriers in the electron transport chain. Now here I have arranged them in order of energy, so descending energy. In a moment I'm going to show you how they are physically positioned alongside each other in the inner mitochondrial membrane. The main complexes are complexes 1, 3 and 4. There is another complex, complex 2, I will put that on in a moment. These are multi-protein complexes, so they consist of lots of protein subunits. So complex 1, for example, it's got about 45 protein subunits in there, and these consist of the electron carriers and various coenzymes, cofactors. The total relative formula mass of complex 1 is almost 1 million actually. I think it's about 980,000. So these are massive protein complexes. As well as the complexes, you have some smaller components. Q is an organic molecule which can move around in the membrane, and it sort of shuttles electrons from complex 1 to complex 3, and also with complex 2 here. Cytochrome C is a small, relatively small protein, a heme protein, so it contains an ion which is in a similar environment to the ion in hemoglobin. If we just take a look at what happens when NADH releases its 2 electrons, they go through complex 1 via Q to complex 3, via C to complex 4, and then finally onto oxygen to give the water. And some of the energy changes, I have indicated them here. So going from NADH to coenzyme Q, we lose 69.5 kilojoules per mole, so that's per mole of NADH oxidized. And similarly, going from Q to cytochrome C, so that's through complex 3, we lose 36.7 kilojoules per mole, and then from cytochrome C through to oxygen, complex 4, we lose 112 kilojoules per mole. So the total amount of energy released by just 2 electrons, well, it's 2 moles of electrons or a mole of NADH, it comes to about 218, so 218 kilojoules of energy. In principle, you need about 30.5 kilojoules to make a mole of ATP. So in principle, you could make just over 7 moles of ATP from a mole of reduced NADH. In practice, we get about 2.5. So if I was to times 30 by 2.5, that would mean 76.25 kilojoules of energy are actually being turned into ATP. So of the 218 kilojoules that are released altogether, 76.25 of them finish up as ATP. So that is about 35% efficiency. If you compare that with a car, normal automobile, that is extracts from the fuel and converts into kinetic energy movement, it's less than 30% efficient. Under biological conditions where the temperature and the concentrations are different, and these values will differ from these standardised values, it's believed to be up to 70% efficient. So it's a remarkable machine, the electron transport chain, for extracting or harvesting the energy lost by the electrons as they pass through the components and storing it as ATP. Now, something that's really important to appreciate is that the ATP is not made at these points. It is not made as the electrons pass through these complexes. This energy, as it's released, it's used to create a proton gradient across the inner membrane of the mitochondrion. And that proton gradient, the concentration gradient, acts rather like a battery. It's a temporary store of energy, and that's going to be released to generate ATP right at the very end by the ATP synthase. Before we look at that, let's just have a quick look at FAD. Now, when FAD feeds its electrons in, not into complex 1, but at the level of Q, but it feeds them through complex 2. So I'll put complex 2 like that. So FAD releases its electrons into complex 2, and then they feed into this Q. So this Q, it's able to move around in the membrane between complexes 1, 2 and 3. So it's rather like a shuttle bus at an airport between the terminals. Just, oi! Come on. We're talking about FADH2 feeds its electrons in via complex 2. So the complex 2, it's associated with the Krebs cycle. In the complete oxidation of one molecule of glucose, you generate 10 molecules of NADH, so 10 NADs get reduced. Only 2 FADs are reduced, and this occurs in the Krebs cycle. And one of the enzymes in the Krebs cycle, succinate dehydrogenase, it's sort of associated with this complex 2 on the inner mitochondrial membrane. The point is, when FAD feeds its electrons into the electron transport chain, as the electrons go into complex 2, the energy released is only about 16.4 kilojoules per mole of FAD reduced, compared with the 69.5 kilojoules per mole when NADH feeds them in via complex 1. So basically you're getting less energy out of the electrons coming from FADH2 than you get from NADH. So going back to our waterfall analogy, NAD is like having water in a reservoir at the top of the hill. FADH2 is like having water in a reservoir perhaps 2/3 of the way up. So the oxidation of FAD doesn't give us quite as much energy as the oxidation of NAD. So in practice, we've said that with NADH oxidation, so per mole of NADH you get 2.5 moles of ATP, or just per molecule of NADH you get 2.5 molecules of AT. Per molecule of FADH2, in practice you get about 1.5 molecules of ATP produced. Again, we've seen already that it's not 100% efficient. We should get, in principle, we could get 7 from NADH, but we get 2.5, and you can work out what we would get from FADH2. In this diagram then we can see how the various complexes and Q and C are arranged in relation to each other in the inner mitochondrial membrane. So this is this blue section here. I've just shown some of the phospholipids to remind you that it's a lipid bilayer, but only for a section. But of course it's all a lipid bilayer. So this inner mitochondrial membrane, of course, it's this blue part here with the cristae. These complexes are located on sections of the inner mitochondrial membrane. So this side is the matrix, which is in here, and then on this side of the membrane we have the intermembranal space. So this black line here is the outer membrane of the mitochondria, so this would be over here somewhere, and we've got the intermembranal space. But remember, this outer membrane is relatively permeable, so ions and molecules can diffuse between the intermembranal space and the cytoplasm. If we're looking at a section of the electron transport chain on one of the cristae, then of course this space here would be sort of within the finger, within the cristae. Right, so that's the physical arrangement, and if we look at what happens then when we deliver a pair of electrons by using NAD, the reduced NAD, then NAD becomes oxidized to NAD+. We get the two protons, two lots of H+, and the electrons are delivered into complex 1. So we get the two electrons going in. I'll put that in red actually. So the electrons come off and they get delivered into complex 1. Now I'm going to show you in a moment that during this process protons end up being transferred across the membrane. Okay, but just for the time being we'll put these protons here because it's more than just two protons that get transferred. But the point is, it's only the electrons that get carried along the chain. There are some positions in the electron transport chain where protons are also involved, but in the main it's electrons only that get transferred. So for example, this one here, cytochrome C, can only carry electrons, so can complex 4, so can complex 3. This one picks up and drops off hydrogens, but really this is the electron transport chain. And finally the complex 4, it delivers the electrons onto oxygen, so oxygen becomes two molecules of H2O. Now you'll notice that, or you'll hopefully remember that we've got 4 hydrogens going on here, so that's the equivalent of 4 electrons. So in fact what happens is that this complex 4, it holds 4 electrons, so you have to oxidize 2 molecules of NADH, send 4 electrons, they sort of build up on here, and then you get altogether, so it would be 2 times 2 is 4 electrons, and they add on to the oxygen, and you would need 4 H pluses, and that's, if you like, the bang at the end. That's when oxygen finally becomes water. Rather than letting the energy out in one go, we actually let the energy out in small steps, as we've seen in the energy level diagram. As the electrons pass through each complex, some of the energy is lost, and that energy is lost by the electrons and it's captured in the form of proton gradient. So some of the energy is used to transfer H plus ions across the membrane. So at complex 1, for example, you get 4 protons pumped across. You get 4 protons pumped across complex 3. Now, strictly speaking, they're pumped by the Q, but in the A-level biology books, that's not really, don't give it in such detail. And you also get protons pumped here by this complex here. You get 2. Now, I wouldn't worry about these numbers. I'm just putting them there because that's what they happen to be, but you're not expected to account for them. To be able to account for all the protons and electrons, it gets way beyond A level. All you need to appreciate is that at these 3 complexes, complexes 1, 3 and 4, as electrons pass through them, the electrons lose some energy. That energy is used to transport protons across the inner membrane into the space. The mechanism by which this happens at complexes 1 and 4, it's a proper proton channel. Over here at 3, it's a slightly different mechanism, and I'll explain that to you later by way of an extension topic towards the end. So what we've done is we've built up a proton gradient, but we haven't yet made any ATP. Now I'll show you in a moment how that proton gradient is finally used to make ATP. If you remember, FAD in the reduced form, FADH2, when it is oxidized, it feeds its electrons its electrons go in to complex 2 and then they go via Q. So you're missing this proton pumping site, and of course you get 2 protons. So because we transfer fewer protons, oops, that's not correct, it's 2 H pluses, because we transfer protons only at 2 sites when we feed electrons in using FAD, then we don't get as much ATP made. So it's like you've got 2 turbines on your waterfall instead of all 3 of them. So let's just have a little look at this proton gradient before we come back to this and see how we make the ATP. Okay, so I'd like you to just imagine that this is the inner mitochondrial membrane, and before we have any electrons flowing through the chains, the electron-transport chain, we have an equal concentration of H pluses in the matrix and in the inner or intermitochondrial membrane space. Always will be protons in solution because water molecules undergo partial dissociation, as the chemists, most of you will know. If we wanted to transfer protons from this side of the membrane to this side of the membrane, well, first of all, we couldn't do it through the phospholipid bilayer, so it'd have to be some kind of proton channel or transport protein. But in order to send protons from this side of the membrane to this side of the membrane, we'd have to have an input of energy. So if we transfer some of these protons, so that one goes across, that one goes across, we're moving the protons against the concentration gradient. So we're building up a concentration gradient. We can also call it a pH gradient. It's actually called a proton motive force, a PMF, which is analogous to an EMF, electromotive force, which is what you have as a voltage or a potential difference whenever you have electrons separated, and that would be just from a battery or an electrochemical cell. So we've got a proton motive force, and that is a store of energy. Now, normally when you transport ions across a membrane against a concentration gradient, you get the energy that you need from ATP. So ATP hydrolyzed to give ADP, an inorganic phosphate, and the energy released from that is used to transfer an ion or a molecule across a membrane. If you think about it, we don't need to do that here because these transporters, if we can refer to them as that, they're taking the energy that they need to do this directly from the electrons as they bounce down that electron transport chain. So we don't need to convert the... this is how we make the ATP. So we haven't made the ATP yet, so we're just taking energy directly from electrons as they lose that energy, going from one carrier to another or through each carrier, and making a proton gradient. Now, if we open up this membrane, so let's put a little channel in here, a proton channel protein, let's describe it as. This is exactly what you have in the inner mitochondrial membrane, by the way. So there's the membrane. Got all the protons on this side, or lots of protons on this side that we've transported there, and relatively few on this side. So if we open a channel through the membrane, so which way are the protons going to go? They're not going to go that way. They're going to spontaneously diffuse down their concentration gradient from a place of high concentration to a region of low concentration, and that is releasing energy. Now, that energy would normally be dissipated as heat through heating, but what happens in the mitochondria is that you have an enzyme associated with this channel, and this is the ATP synthase. This is the enzyme that's finally responsible for making ATP. So it's ATP synthase. And what that does is say ADP plus inorganic phosphate and generate ATP. Now, to do that, you need to use energy. You need an input of energy, 30.5 kilojoules per mole of ATP made. The energy is taken directly from the collapsing proton gradient. So just like we put energy in to pump the protons across the membrane, when they come back they release energy, and that is sort of tied in or coupled to the synthesis of ATP. So this is the actual phosphorylation step. We're phosphorylating ADP to make it into ATP. That's the phosphorylation part of oxidative phosphorylation, and it's called oxidative phosphorylation because this is tied in or coupled to the oxidation of NAD and FAD, or you can even think of it as being tied into the oxidation of glucose ultimately. If I just zoom out again. Okay, so I can put one of these proton channels in the membrane. There are lots of these on the inner mitochondrial membrane, so there is the proton channel, and I better show that we've got lots of H pluses on this side because they've been transported across. And so they're allowed to flood through this channel. So the H pluses, they can then come back into the matrix, and then they are used to make water. So they're getting reunited with their electrons. They parted company really. The protons went into the intermembranal space, the electrons went down the electron-transport chain, and then the electrons come off at complex 4. Protons come through the channel, they reunite, so 4 protons and 4 electrons gives 4 hydrogen atoms. That makes your water. And of course the ATP synthase is sitting here on the inner mitochondrial membrane, and ADP plus inorganic phosphate makes ATP. And as I said in the previous diagram, each mole of NADH gives 2.5 moles of ATP, or each molecule of NADH would give 2.5 molecules of that. Each molecule of FAD would give only 1.5 molecules there. Something really important is it's called coupling between ATP synthesis and the oxidation of DH and FADH2. Electrons can only flow freely through this electron transport chain to oxygen if at the same time ADP is being phosphorylated to ATP. So if your cells have got lots of ATP in there and very little ADP, so you're resting for example, that ratio of ATP to ADP being high will inactivate this complex 4, so electrons will not be allowed to flow onto oxygen. And because that one is not operating, these will all stop. It's rather like a traffic jam. These are like cars coming down a road. If there's a road jam here, you get a backup of electrons. So electrons will be sort of parked on all of these electron carriers. If you start exercising, your ATP is used in muscle contraction, so the amount of ATP decreases and ADP increases, so you've got a high ADP to ATP ratio. That allows this complex to be operational and electrons can flow. So the point is that phosphorylation and oxidation are coupled, they're tightly associated with each other. There is such a phenomenon as brown adipose tissue or brown fat. I think human beings have varying amounts of it. Hibernating animals, they have brown adipose tissue which they use to warm themselves up again when they're coming out of hibernation. So when an animal is hibernating, its body temperature is very low, and when it needs to warm itself up again in the spring to wake up, it has some proton channels just like these, but they don't have the ATP synthase there. It's called uncoupler proteins. They allow H plus ions to flood down the concentration gradient back into the matrix, but the energy isn't used to make ATP. Instead, it's just dissipated as heat and it warms them up. Just going to show you how the proton pumps at complexes 1 and 4 are believed to work. The proton pump at complex 3 works in a slightly different way. This is the inner mitochondrial membrane. So we've got the matrix here and the intermembranal space here. And these black blobs, they just represent the massive protein complexes, either complex 1 or complex 4. So if it's complex 1, it's got 45 subunits. But going through that complex, there is a channel, so it provides a connection between the matrix and the space between the membranes. And we have some water molecules associated with the they're hydrogen bonded onto the amino acid residues, the side chains of the amino acids in the proteins. I say this is very schematic, a stylized interpretation. Here we have a proton, and it's in the matrix, and we're going to transport it across in the other side of the membrane. When you have protons in water, they occur associated with water molecules. So if you're doing A-level chemistry, you'll know already that you have a water molecule there, and a proton attaches itself using the lone pair, so it makes a dative bond to the hydrogen. So you get a H plus attached onto the water molecule. So you'd normally write that as H3O plus, the oxonium ion. So don't worry about this if you're not doing chemistry. It's just that protons attach themselves to water molecules. And what happens during the proton pumping is that the proton attaches itself to one of the water molecules that's lining this channel, and then it can hop across onto another one, and then onto another one, and then out on the other side. Now maybe places in here where it's not actually a water molecule, it might be exchanging with a proton on an amino acid side chain. This is all relatively new research, I would say. It's not fully understood what happens at the two complexes, certainly not so much at complex 4. But that's basically the means of transferring the proton. So the waters are acting as a proton wire. Protons are very good at hopping from one molecule of water to another. It happens in water. If you just got a water solution, you put a bit of acid in, the protons hop around by hopping from one water molecule to the next. And that's exactly what's happening when this go through this protein channel to the other side of the membrane. Now, how is this associated with electron transport? Well, you've got your electrons coming into, let's say this is complex 1, so the 2 electrons are coming off NADH, and they're going through the system and off to the Q. As the electrons go through the protein complex, they cause it to change shape. So these proteins change their tertiary shape, the tertiary structure, depending on whether they're in the oxidized or the reduced state. Well, it's not really the proteins that are in the oxidized or reduced state, it would be the prosthetic groups that they have. So you've got ions in some of them, for example. The point is, when they're carrying electrons in the reduced state, they're in a different shape, three-dimensional shape, to when they're not carrying electrons, when they're in the oxidized shape. And you can think of that as either opening a channel at one side or closing it at the other, depending on whether electrons are on the proteins. More chemically, you can think of this as changing the pKa's of the amino acid residues, or where the protons bind. So if you've got a high affinity at this side, it means a proton binds, and then it lowers on this side and it comes off, and that's all controlled by the redox or the reduction level of the protein. Now at complex 3, a slightly different mechanism operates. Okay, I'm now going to explain to you how proton translocation occurs at complex 3. I think translocation is a better description than proton pumping. You'll probably see why for yourself when I explain it. First of all, it doesn't really occur through complex 3, rather it's the interface between mobile carrier, coenzyme Q, and complex 3. The thing about coenzyme Q, it's a quinone, and that means rather than carrying only electrons, it can accept a whole pair of hydrogen atoms. So we put 2 hydrogens there, and that gives us the QH2. So in the reduced form, the fully reduced form, it's QH2. Now, I can represent that on here. So initially, Q is sitting at the surface of the mitochondrial membrane facing the matrix, so it can pick up 2 electrons and 2 H plus ions from FAD. So FADH2 goes to FAD, the oxidized form, and it transfers the 2 electrons and the 2 protons together. Remember, that's just the equivalent of 2 hydrogens onto Q, and then Q diffuses up towards complex 3, and complex 3 accepts the electrons from this, but only the electrons. It doesn't accept the protons because the initial acceptor in complex 3 is an ion. In fact, it happens twice and it has to go through various cycles, but basically ion can only accept ion 3 going to ion 2. It involves only electrons, no protons. So the electrons get loaded into complex 3, but the protons don't, so they then just release. And by this time, because the CoQ is closer to the other side of the membrane, the protons get released into the space between the membranes. So Q has picked up 2 hydrogen atoms, the electrons have gone to complex 3, the protons have been released into the space between the 2 membranes, and then it just diffuses back again. And in fact, it does, for every FADH2, it actually does 2 cycles. It's a lot more complicated than this. The electrons are transferred one at a time, and you get the half-reduced version of this called the semiquinone, which does a loop as well. And of course, Q is also reduced by by the complex 1. Well, you've got complex 1 over here. That can send a pair of electrons onto Q. Those electrons came originally from NADH, and when that happens, it picks up 2 protons from the matrix, and so that it becomes the fully reduced form, and then it releases the electrons to the complex 3 and the protons into the space. So it's called the Q cycle. It's responsible for pumping protons at complex 3. Just going to give you a little idea about how the ATP synthase uses the proton gradient to generate ATP. So the blue part is the inner mitochondrial membrane, just represent it like that. I've got these protein complexes associated with the membrane. So on this side we've got the matrix, and we have lots of protons on this side of the membrane, so that's in the intermembranal space. We're going to allow them to flood through this channel protein to this side, and as they do so they're losing energy. We're going to see how that's used by the ATP synthase to make ATP. Now the way it works is the protons come in and they attach to a binding site on this part of the enzyme. It's separate to, think of it as being slightly separate to this part of the protein complex, so I'll just colour that in. This is all highly simplified and schematic, and it's just to explain, give you an idea of the mechanics of it rather than exact mechanistic details. The proton, when it binds to its binding site, it finds itself next to an amino acid residue on another part of the protein that's positive. So this green part in the middle is like a bobbin wheel. It rotates, and so that proton finds itself slightly rotated round relative to this one. So this whole thing, if you're looking down on this, it's actually going to rotate. Okay, and so basically protons, they get taken around and then they come out on this side back into the matrix. So it's based on wherever a proton binds to this protein, it finds itself repelled by a positive charge on another bit of protein, so it makes it move a little bit, and every time one adds on, it results in the overall rotation of this part of the protein. And this part of the protein, it's got a little sort of, it's called a cam, just like a camshaft on an engine. So it's not symmetrical. So as this rotates, this bit wobbles. So if you look down on the top from this, it would look like that, let's say, and this bit rotates round as the protons move through this channel. So we're turning a proton gradient into a rotational movement. So we're just going to now have a quick look at how rotating this lobe or this cam, how it interacts with the ATP synthase and allows the ATP synthase to make ATP. This diagram represents the ATP synthase protein. So if we had the proton channel, what we just looked at would be something like that, and the ATP synthase, excuse me, would be sort of sitting like this, and we're looking from underneath, we're looking up at it. So this green protein, which functions as the channel for protons, that's sort of interacting with this one, and we said that the bit that turns round is sort of lobe shaped, so we say it's a cam, and it's going to rotate around, so it's going to go around like that with it, and whereas this black part, the ATP synthase protein itself, stays still. So it's like there's a little shaft there, and this just goes round and round. As it goes round, it squashes the protein in different places. So the ATP synthase has got three sort of active sites, if you like, and each one gets squashed by this lobe as it goes round. So there's always one site that's empty. We've got one site which is open, we've got another open site, and on that open site ADP attaches and inorganic phosphate attaches, but they're not close enough together to react and for a reaction to take place between them. But when they get pushed really close together like this, so you've got the ADP there and you've got the inorganic phosphate there, that's when the reaction takes place, and they make ATP, which then is released, and then you go back to the other side. So remember, this is rotating around, so what will happen when that cam has rotated around, then we get to this site and some more ADP and inorganic phosphate binds. So this cam rotates around, it squashes this, brings these together, and then they will then release ATP, and then it brings these together and they will release ATP. So it's just a very clever rotating device which squashes different parts of the protein to bring the ADP and the phosphate together, and when it relaxes, out comes the ATP. So that's a highly simplified version. I could give you I could spend a very long seminar just explaining how that works. And it's probably a bit too complicated to do on here. It's just to give you an idea that the whole of the mitochondrial electron-transport system, it's a machine for, just like a car engine is a machine for getting mechanical energy out of the chemical energy stored in the petrol. Respiration is a machine, if you like, for getting the energy, the chemical energy out of glucose and turn it into another form of chemical energy, but in smaller currency. So it's like, as I said in the video on ATP and glucose, glucose is like a £50 note, ATP is like having a pound coin, it's the loose change. So we're breaking down the glucose to make lots of ATPs. And this is what's happening, this is how it happens, but it actually goes through some mechanical steps. We go through an electrochemical gradient of the protons, and we go through this mechanical device which is driven by the protons collapsing down their gradient and finally affecting the shape, the 3D shape of the active site of the ATP synthase such that ATP is generated. Okay, so as I say, most of that is extension material, this last few pictures, so you must check on your in your textbooks and your specification, depending on which examination board you're following. But maybe bits of this you can put them into your synoptic essays to get the higher marks. So I hope you found that useful. If you've got any further questions on aerobic respiration, then by all means use the comment section below. Thank you very much.